Okay, so this is video 3, and in this video we're going to be looking at aerobic respiration. So following glycolysis, if there's oxygen available, we will utilize aerobic respiration and our mitochondria. But in order to understand aerobic respiration, we have to understand mitochondria structure. So when we look at the mitochondrion, it actually is a double membrane organelle. And the reason for the two membranes is based in the endosymbiotic theory. So I highly recommend looking into that more. But anyway, so when we look at our mitochondria, we have two membranes. We have an outer membrane and an inner membrane. And on the inside, we have the matrix. Now, the matrix is where the citric acid cycle or the Kreb cycle occurs. Now, our intermembrane space is where we'll have a proton gradient accumulate, which is super important for establishing like a difference in charge across the membrane that we'll talk about during chemiosmosis and oxidative phosphorylation. Fun stuff coming up. But anyway, so we have an inner membrane and an outer membrane. And when we talk about what's happening in the inner membrane, so our electron transport chain that we'll talk about is located within this inner membrane. And the inner membrane, when you look at it, you can see it's highly convoluted, or it's highly folded. Now, the reason for those folds, or that those folds have evolved, is to increase the surface area to host or to have space for more electron transport chains and ATP synthases. So the more highly folded it is, the more ATP can be produced. Okay, so this is the overview of the mitochondria's structure, and let's go ahead and see what happens. So at the end of glycolysis, we ended up with a net gain of 2 ATP that will be used by the cell for energy. And now we have 2 pyruvates and 2 reduced electron carriers. So all of these will move into the mitochondria, where they'll be utilized for aerobic respiration. So let's think about the big picture though for a minute. So we started, let's see if we can make this larger. So we started with a 6-carbon glucose. Now, in glycolysis, glycolysis literally means the splitting of sugar, glycolysis. And so we start with that 6-carbon glucose that is broken into 2 pyruvates. Each pyruvate still has 3 carbons. There's still potential energy in these bonds. So now that we're in the mitochondria, we need to harvest the rest of that energy. We need to fully oxidize our food. And when you think about what oxidize means in terms of this process, when we oxidize our food, we're going to take the electrons from our food. And when we think about, well, what are electrons in a cell? When we move electrons in a cell, that's how we move energy. So basically, when we harvest those electrons, we're getting energy from our organic molecules. So with these 3 carbon pyruvates, there's still potential energy in them. So between the oxidation of pyruvate and the citric acid cycle, between those 2 steps, our food will be fully oxidized. All of the potential energy that we could possibly gain from our food will be in the form of reduced electron carriers. Okay, so that is the main purpose in the next 2 steps. So let's go ahead and look at how we do this. So our first step in oxidizing our food even more is called the oxidation of pyruvate. And there's basically 3 things that are going to happen here. In the chemical reaction that occurs, when we oxidize our food, we're going to take those electrons. And so if pyruvate is oxidized, that means that something is going to be reduced. And in this case, the electron carrier, NAD+, is reduced, forming NADH. So here we have a reduced electron carrier that will carry those electrons to the electron transport chain. And when we see pyruvate having 3 carbons, Well, when we just broke that bond and took those electrons, one of the carbons came off in the form of carbon dioxide. So when you were actually exhaling your carbon dioxide, the carbon dioxide you breathe out used to be part of a glucose molecule or part of a lipid or part of a protein. Like when you exhale, your carbon dioxide was somehow incorporated into your food or into your body. So for example, well, we'll talk about that a little bit more after the citric acid cycle. Okay, so far we have a reduced electron carrier has been formed and a carbon dioxide has been made. Now, carbon dioxide is nonpolar. And because it's nonpolar, it is able, now we need to remember, the mitochondria is membrane bound, which means it's a lipid bilayer. And so this right here is a lipid bilayer, and the outer membrane is a lipid bilayer. So that nonpolar carbon dioxide can diffuse right on out of the mitochondria and even right on out of the cell. Okay, so here our third and final step of the oxidation of pyruvate is a coenzyme called coenzyme A that will attach to our acetyl. Now, co means to help or work together, so this coenzyme A is going to help the acetyl basically join in or start the citric acid cycle. So there goes our nonpolar carbon dioxide. And so let's go ahead and see how the citric acid cycle is going to work inside of the matrix. So this so this is all going to happen twice because we still have a whole nother pyruvate from the end of glycolysis. Okay, so we're going to look now at the citric acid cycle, and here we go. So the citric acid cycle happens within the matrix, and you can see over here we already have 4 reduced electron carriers made, 2 from glycolysis, 2 from the oxidation of pyruvate, and now we have our acetyl-CoA. So the Kreb cycle, or the citric acid cycle's main purpose, is to fully oxidize our food, to get all the potential energy out of our food into actual energy that's usable, right? So that's what we're hoping to see by the end of the citric acid or Kreb cycle. So keep in mind the citric acid or Kreb cycle is a metabolic pathway. It's a series of 8 steps, all controlled by enzymes. And if you are an AP Bio, we don't need to know or memorize those steps, just kind of understand the big idea. So here we go. Our purpose, okay, here, all things I've just mentioned. So now let's go ahead and see the first step. So an enzyme is going to take that acetyl and attach it to a 4-carbon molecule called oxaloacetate. Now, this reaction occurs and forms citric acid. So that coenzyme A was just like a helper to help this reaction occur. So here, the first molecule formed in the citric acid cycle is citric acid, and it's a 6-carbon molecule. But it was really that acetyl-CoA, the acetyl that attached to the 4-carbon oxaloacetate. And so since this is a cycle, we're hoping that after all 8 steps, we end back with the 4-carbon oxaloacetate. So we can start all over again. Okay, so here we have citric acid, and basically we need to fully oxidize our food. So as an enzyme comes and oxidizes, so it's going to break the bonds and take the electrons, it's going to reduce an electron carrier. Now you notice that carbon dioxide left because it's nonpolar and it's what we exhale. So again, one of the carbon dioxides left, and we are left with a 5-carbon molecule. So again, another enzyme is going to come and it'll oxidize it, there goes a carbon dioxide, and reduce an electron carrier. So at this point, my sweet students, we started with 6-carbon glucose, and then we, in glycolysis, broke it into 2 pyruvates. And then we'll focus on just 1 right now. And so during the oxidation of pyruvate, one of those carbons came off as carbon dioxide, and we're left with acetyl, a 2-carbon molecule. And then one of those carbons were oxidized from our molecule and came off as carbon dioxide, and then again carbon dioxide. So at the end of the citric acid cycle, your entire glucose molecule is now in the form of carbon dioxide being exhaled. Now, why I think this is beautiful is because while we're using glucose as our example to teach this, we also burn through fat in the same way. So our fatty acids that we store actually are broken down and oxidized during this citric acid cycle. So if a person loses 10 pounds and you wonder, like, if you think about the conservation of mass, like mass isn't created or destroyed. So therefore, if you had 10 pounds of C, H, and O in the form of fatty acids on your body, and all of a sudden you've lost weight, like it didn't just magically poof and disappear, you literally exhaled it. So as you broke those bonds in the fat to harvest the energy, you exhaled the carbon dioxide, and that's where your matter went. So as you're running down the street, exercising, losing weight, you're actually exhaling the fat from your body. Anyway, I think that's beautiful. Okay, so back to the citric acid cycle. So now you see we have even more reduced electron carriers. Now in the process of the citric acid cycle, we do get one ATP produced by substrate level phosphorylation. However, to be truthful, it's actually a GTP that's made and then converted into ATP. And then there's actually a few more steps involved in the citric acid cycle that we, again, don't need to know. But basically, you're going to have, there's another type of electron carrier called FADH2 that will be oxidized and not oxidized, I'm so sorry, will be reduced to form FADH2. And then we get one more electron carrier is going to be reduced, forming NADH. And now look, we're back at that 4-carbon oxaloacetate, so we can start the process all over again. Now, I want to point out that the citric acid cycle actually is a very complicated process with lots of substrates and enzymes. And this was it's a very general overview. So I know I was vague in my discussion. Feel free to open a textbook, go on Google and study these steps. But as far as AP biology is concerned, you don't need to memorize them, okay? So that was all for one acetyl-CoA, but if the next acetyl-CoA comes in, because remember, we had 2 of them from one glucose, and the process goes all over again, we basically will make 2 more carbon dioxides, as well as a bunch more reduced electron carriers. So by the end of the citric acid cycle, we have fully oxidized our food. We have broken it completely down into carbon dioxide that we exhale. And we have in the process reduced a whole lot of electron carriers that can move those electrons over to the electron transport chain. And I think I'll stop my video here and start a new one for the electron transport chain and oxidative phosphorylation.